It's been an absolute pleasure. The team have looked after me wonderfully well. It's great to be in Brussels. It's not a city I know well, but my daughter has decided to settle here, so it's been wonderful to also meet up with her today. So yeah, you're going to stop me when I've spoken too long, because I have too much to talk about. What I'm going to start with is the story of where this came from, which as working with and as effectively a zooarchaeologist looking after a student who's a vet interested in organic farming and the way that was different in the past. He was working with animal bone. And the discovery that he was exploring, and the question he was asking was, in the end, because he needed to get animal bone where we had the whole jaw, so he could look at tooth eruption and therefore get age, and he needed to get sex, which we got with DNA, to answer the question of how different populations in the past managed their animals. And the question he posed himself was, do you see differences in the way that on the west coast of Scotland, there is a difference between the Iron Age and Viking farmers? And what he was doing was he was comparing that very interestingly with organic farming practices. And the result you can see here is if you look at your organic dairy practice, the numbers of animals of the different ages that you kill, and then compare it with your Iron Age and Viking sites, what you see is that the Iron Age looks more like a dairying practice, and the Viking sites looks more like there's a more continual slaughter, and also a predominance of male animals, suggesting probably that this is actually for meat predominantly. But the real story of this paper is how few data points there are. There was so little data we could get of whole jaws that these scales are here, 5, if I can point this out, 5 here and 7 here. So it was very hard, even though there's more than 4,000 animal bone fragments, to get enough jaws to really answer the question statistically, which was really frustrating after 3 years. And so, yes, maybe the Vikings didn't have much interest in milk, but the reality was, when you're sorting through all of those animal bones to get that kind of question is difficult. So you can imagine the day I was taken into the Borthwick Archive in York and saw this. Boxes after boxes of parchment. And what is parchment but the skin of an animal with, if it's a legal document like these, the date written on it. So suddenly going from like 29 jaws in a whole project in 3 years, you're looking at thousands and thousands of dead animals with the date written on them. How much could we learn about this incredibly rich archive? So that's what I set out to do. It's a library of livestock management, if we look at it as a biological tissue and not as a medium for text. And so what we discovered early on, this is Sarah Philippe's work, who really was the founder of the field of what's now called biocode ecology, is we got funding for Sarah to come over as a Marie Curie Fellow. She joyfully went into the library on the first day that she visited the archive. And she said, I'm going to take samples of all of your parchment. They said, no, you're not. You can't physically take samples. It's like, what? And I remember her coming back to the office going, no, I don't know what I'm going to do. And so what she did, which was very smart, she sat with the conservation team in the conservation studio, and she analysed every bit of waste that they were generating from the different types of conservation. And she made this great discovery. When they were cleaning the surface of parchment with a PVC eraser, a Mars Staedtler eraser to get rid of the dirt, when they threw that eraser waste away, it contained proteins, and later we discovered also DNA. So that meant that a standard conservation practice that they were doing, they were throwing away the eraser waste rubbings at the end of the cleaning. If we just put them into a tube, we could suddenly get the collection of parchment from libraries and archives all over the world. And we met up with a very young conservation student at Norfolk County Record Office who very kindly wrote us the text which explained how to do this as a conservator. And then we put a video on YouTube which explained how you do the work and I am not showing that now, so that we got the idea out. I went to archives and conservation meetings, we talked to lots of people, And by the end, we had over 140 libraries and archives all across the world, feeding samples back of the waste that they were generating from conservation practice. So it really was at the very beginning, it was very much a joint project. And this is the technology we're using at the time, which comes out of a wonderful project with Vincent sitting there, who is in charge of the European Animal Feed Institute for qualification. And we work with him. We come up with a method he didn't choose to use, we call zooms, which enables us to take very small amounts of the protein collagen, the skin protein collagen, which is the dominant part of parchment, digest it with enzymes and get a fingerprint. And that's all you need to know. It'll fingerprint and it'll tell you what species you have. So that's the science. So when you think about it, this is basically how it works. These different masses represent peptides. You can look at a series, these are a series of historical charters, again, dated documents, and you look at certain markers. This marker here says it's got to be above it, this 1105, and if you've got the 3034, it's a sheep. And if you haven't got that, it could be a sheep or a goat. It can't be a cow. So you can, by looking at these simple fingerprints, very quickly, in a second, identify what the sample is from the eraser waste from conservation practice. And therein lies the story.
But
so now,
watch a very old idea taking shape. At this factory, craftsmen have been making parchment and vellum for centuries. These are the lime pits for cleaning the calf or goat skins before the hairs are removed. And now comes the unhairing process. In this case, a calf skin from which vellum is made. Goat skin is also used for vellum, and sheepskin for parchment. The skin is tightened on a frame, part of the process that has remained unchanged for at least 2,000 years. And some of the Brentford workers have had a good innings too. The semicircular shaving knife is a tool that's peculiar to the craft, and no other would be so effective in leveling the skin. But even then, only an experienced workman knows how. The skins are given a hot bath and a thorough scraping to remove the grease, and then they're taken to the sheds where they're stretched on frames to dry.
okay, so you've got, this is a very, very rare film now of a practice which is almost lost. The few people still to make parchment are actually not using the same traditional methods. And we have to go back to a master's thesis from 1954, if I'm correct, yes, where in this thesis by Saxel, she actually goes to one of these early parchment sites and she describes the process of production. And as you learned, our project was called Beasts to Craft. We were interested in the animals and how those skins became turned into the writing medium. What could we learn about them by being scientists and studying them with craftspeople and historians and learn what we could? And so the first thing I'm going to ask is the question, and this is from her thesis. She says here, the parchment is made from domestic sheepskins. So you don't have to use sheep. In this particular factory, they were using sheepskins. Today, almost all the parchment is made from sheepskin. Not all, but most. The question is, what can we learn from those sheep and other animals? Well, there's two other species commonly used, calf and goat. We can also ask questions about how old were the animals when they were killed, what sex were they when they were killed, and how healthy were they when they were killed. Can we tell that information from the parchment itself? So, going back to my point, which I've already laboured a bit, this is my student Laura Viñas's work, where she collected together all the archaeological reports she could find on zooarchaeological remains in Spain. She was interested in medieval periods and the rise of merino sheep. This is what you can get from bones. Notice you don't have sheep and goat together, you have sheep-goat because the bones are ovicaprid, you can't tell them apart. In the course of 1 year, just going to libraries in Spain, she could build a picture like this because she can identify, of course, to much higher temporal resolution because the documents have the dates written on them. And she can also tell apart most of the time sheep from goat using zooms. And what she found out here was, of course, that this is a medieval wool economy, so the vast majority of the animals that are being used are sheep. So just in 1 year, she's getting more data about the use of animals than you're getting from multiple zooarchaeological studies, which is quite interesting. And the other thing about this, which is intriguing, this is Laura here, is when you look at the sheep, there's a very important change in the middle of the 13th century in terms of sheep management. And we have these very, very long sheep drove roads in Spain. And you might think this is interesting or perhaps not. What becomes really intriguing is you lay on the human genetics of the Spaniards on top of that, and what you see is that the genetics also follow the drove roads, because these are the kingdoms, these are points of connectivity. So the animals themselves are also beginning to reveal stories about us too. So when we start to analyze these very large numbers of samples, what do we learn? Well, just think about it in terms of where the animals are. If you look in England and Spain, see the sheep there, lots of sheep, big wool economies. Goat, mainly in the south, and then cattle, huge numbers of cattle in Belgium and France and England. So you have these different resources that you could use, and I won't go into the great detail of it, but we've shown that pretty much any style of parchment can be made from any of these species, and that's what people tended to do. So if you look across the different types of documents in the different kinds of countries, surprise, surprise, in England, a lot of this yellow, which is sheep. In France, a lot of the blue, which is cattle. The one that's most interesting, I guess, is that we see a lot of goat in Italy, and that's because the Vatican would always write on goat skin. So it became a tradition in Italy that the best quality skin in Italy is goat. So we can see these patterns of use, which is not too surprising. And then you can ask questions historically. Why is it that goat, which here, from Master Fitzherbert is saying for the Book of Husbandry, is actually a really good animal? Why don't you get so many goat in England? And the answer is down to the laws behind the ownership of goats and the damage they cause. I mean, they're just not worth owning. They're too expensive. So you're seeing the relationship between the social interaction and availability, even though, as he's saying, you should be looking after goats. They're very good. You just don't take them. When we think about the animals themselves, we can get clues about the animals, weirdly, from the way the parchment is being made. And here's Jerzy Vucnek, who's a key member of the team because he is a parchment maker. He crafts parchment. And this was very important. And you'll see some images from, we ran, I think, 6 parchment workshops. We got people working on the manuscripts to make the skins to learn about them. And so one of the things that you do, as you saw from the video, you're now all experts, is you put the skins into a bath of lime, you remove it from the lime, the alkali in the lime is reacting to liberate the hairs, which you can then scrape off the skin on the so-called beam. And if you know anything about leather production in the UK, it's called the beam house. This is the beam. There's a picture of Jerzy scraping away on the beam. Now, this is chemistry. And you're putting skin into lime. The longer you leave the skin in the lime, the more you're going to degrade the parchment. And so you need to target something simple. And so what we did is we targeted the hydrolysis of an amino acid, one of the amino acids, this amino acid called glutamine. And what we were doing, we were measuring, because we use mass spectrometry, a mass shift. When that converts to glutamic acid, that happens in the lime. The longer it's in the lime, the more modification you get. And we called that glutamic acid, so glutamine is Q, the letter Q, the amino acid Q. So we conveniently call this the PQI, the parchment glutamine index, which everyone calls the parchment quality index. But then you talk to scribes, and you can't really use the word quality, but PQI. And this is what you can see here, is that high values indicate very little time in lime. And you see that calf skin is the one that most often doesn't spend so much time in lime. The hairs come out very easily. And then goat skin's more variable, and sheepskin too, and you can see that those are the numbers that we can't identify from this large-scale study from mini libraries. So you can see a pattern individually. And then you can start looking at these, because these are dated documents, right? So you can start looking through time. And what you see early on is that we've got these very high quality, high values in sheepskin. This is indicating, and this is something that's a mystery to us today, how they were able to make very high quality, very fine sheepskin with almost no damage of the skins themselves. This is France with a lot of cattle, and here we have Spain, and we see the same thing, where we have this lot of initial, early stuff is very well made, never see it again. It becomes a different style of production, which then we can't recreate today. And then this is Italy, all of that goat skin that we're seeing. And then in England, we don't have such early material, but this massive amount of sheepskin. And intriguingly, the pattern of fall off between the 12th and 15th centuries is almost identical between England and Spain. They must be also following the same practices of production. So you can see these kind of big scale pictures of things. But then, think about the production itself. The lime was useful. That told us about the way they're making the parchment, how that changes with time. What else can we learn? So you saw this before, and this is us and one of our parchment workshops, scraping. This is David, and this is Eri's colleague, and we're just scraping the parchment. When you look under raking light, you can see the work of the parchment maker. You can see the scrape marks on the skin. And this is telling you about the way that they are viewing the parchment. And so often you get very beautifully written text on some pretty ugly skins. But the other thing you can do is you can unite the edges and you can build back the animal from the book. Okay? So here we have an example where we've managed to rebuild the animal out of the individual folia. So each of those, the animals have been cut into 4 pieces. They've been folded over to make folia. We can, using imaging with backlight, and we can virtually reconstruct the animal. And now we can start looking at the size of the animal. How old was it when it was killed? And so here we have an example. We've taken a book, and we've literally taken the whole book, and we've worked out how they have made the book from cow and sheep. And one by one, we can bring the animals back out of the book. And we can learn the way that scribe, or actually 2 scribes, there's a whole story about a joke in there, which I can tell later, the scribal joke buried in that book. But you can see here that when you construct the skin, you can then measure the length, and you can also look for proteins in the parchment, which tells you in the case of calf skin, they've got proteins which are circulating in the fetal and young animal blood. So we know these animals, because they do change in size with time, and of course the skin gets stretched. They're being killed at between 6 and 8 weeks of age. That's really, really young. And this is the size of one of those calf skin. This is Zooarchaeology, very convenient in York University, and that calf is the right size. It's actually quite small for a modern calf. And so you ask the question, what does that mean to a medieval economy if you're killing calves that young? Well, here's a picture. This is a picture of a cow being milked, and notice she has her calf with her. They needed the calf with many of the breeds they had, or the types they had, not breeds, to have the letdown response to release the milk. And to kill an animal that young is actually going to impact on your milk production. So this is Walter of Henley, and what he's saying is that you sort of take 1 teat every week away from the calf, so that milk becomes yours, and the calf is being slowly weaned off the milk. When you kill the animal so young, then you are wasting production, because that animal could grow on to be a much bigger animal. One thing you are getting, though, by killing that young calf is you're getting the stomach contents, and in the stomach you have an enzyme called rennet, and rennet is important for making cheese. And so it's kind of interesting to think about this, is that 1 young calf skin could make 50 pounds of hard cheese, and that hard cheese was a very important tithe in medieval times. So perhaps one of the virtues of killing these calves was the fact that it would then release milk for cheese. Then another question is, why are you killing them? What sex are the animals that you're killing? Now, early on, when we look in the 8th to 9th century, what we're seeing is they're mainly killing young female calves. But to be honest, the new data we have in the last couple of weeks, that pattern is actually kind of broken down. It's now about 50-50. But of course, in that period of time, arguably castrated male animals, oxen, which were very important for traction, for things like ploughing. At that time, you didn't have the harness to allow horses to plough. So probably male animals are quite important. And we do see very large numbers of female animals being killed. Interestingly, the largest number of female animals we see being killed are in the most highest status texts. And perhaps that's telling us something about God knows that you're killing the very best animals for him. But we don't know that yet. We need more data to understand that better. When you come to Paris, and this is 1539, precisely to the year, Great Bible, at that point we've just got some new data, literally just through, and at that time it's almost all young male calves that are being killed. And that's not surprising because by this time it's a hugely important dairy economy in Paris. So you're killing the male animals because you want the female calves for milk and for cheese. So you can start to look at the pattern in the parchment to start thinking about the slaughter of young animals, the thing we were trying to do before, but now we can do it to the year in different parts of the world. What's really curious, though, going back to those bones, is just the bones are not telling us the same story. Look here. This is showing us not many samples. Again, this is a problem with the zooarchaeology. But look at those blue juvenile columns there. Those are young animals. They're not actually as young as the animals that we are killing. And notice, in the Norman period, where we see huge numbers of calfskin parchment text, they're not finding any calf bones at all. But this has been noted previously. If you look in Doomsday, he says, then what you find is that Doomsday is talking about very large numbers of sheep and very few cattle, and yet this is not calf, this is cows here, but on rural sites, we're seeing about 50-50 from the zooarchaeological record. And this is the challenge of interpreting these different records accumulated in different ways. But it's pretty clear that we are seeing vast numbers of extremely young calves being killed, and yet we almost never find their bones zooarchaeologically. And I do not know what that means. Where those bones are going, I have no idea. But we see them. When we then look at the parchment, this is an interesting piece. It's got a very large tear here, which is able to be repaired. That's fine. But it's got all this at one end. What's that? Well, I don't know. It could be warblefly. Because warblefly enter in the rear end of the animal, and then they eat their way through the calf, and then they come out around the neck, and you see that here. But these animals are very, very young, and this takes a season, so I do not understand that data. But maybe there are clues in the parchment to the physical clues to disease. And so my colleague, Annelise Benoit, at Sorbonne, has been interested in studying the patterns of outbreaks of disease from historical records and from zooarchaeological records. So she's built up this table exploring the data. And we have been unbelievably excited in the last few days to start generating this data, which is confounding, confusing, and extremely exciting. What we're seeing is the wrong species of animal disease on the parchment. We're finding a pigskin disease on sheep parchment, and we're finding sheeppox on sheep and on calf parchment. So what does this mean? That's why I showed you the video of those old chaps making parchment. That's why I showed you the skins going into the lime pit. My guess is what's happening is you're picking up all of the pathogens which are in the slaughterhouse as these animals being processed either for leather or for parchment. It's all going into the lime, and then when they go into the quicklime, the DNA of these animals has been cut to bits, and remember, they took out the parchment, and it's dried onto a frame, and all that DNA from the lime pit is now recorded in the parchment. I think this is a signal of the lime pit, not of the animals themselves, which is incredibly exciting, because what it means is you're sampling basically the livestock management for a small region, a small manorial court, for instance, a few hundred acres, in real time, year on year on year, in every part of the country. And so this is our data. Please don't share this. This is brand new data. But this is sheeppox. We cannot believe we'll get these viral pathogens. We're getting these sheeppox viruses. We've got them from historical analysis, collected all these historical samples here. We've got some Bronze Age material here. But look, we're getting individual parchments from dated documents in medieval Ireland. And we're tracking the evolution of a virus in real time from year to year. This is kind of mind-blowing resolution that we're going to be able to get with this. This is just coming through now, and we're going to start doing more of this. So, when you get disease, you get diseases when your animals are sick and they're immune compromised. When does that happen? It generally happens when you do not bring in enough fodder for them over winter. Okay? So it matters how dry or how wet the year is. So rainfall matters. And there's been a huge amount of work done on looking at the historical records from manorial accounts and thing called manor court rolls. If you don't know what these are amazing documents. So every manor, which is about 1,000 hectares, is the biggest manor. Some are slightly larger. So 1,000 hectare parcels of land. You had the lord of the manor, and every 3 to 4 weeks throughout the year, he would hold a hearing, and then a scribe would write down in real time all the decisions of the court. And once you'd completed one sheepskin, you'd sew on the next one, and the next one, and the next one, for year after year after year. So we have records from manorial courts all across Britain at sub-annual resolution. Okay? And of course, in a bad year, the manorial count at the end of the year, so many animals died. The price of grain went up or went down because there's a surplus or was a restrictive amount. The value of salt was high, was low. So you're getting all this information about the function of the court, and also who has stolen money from whom, or what has happened, what violence there was. So you can look at the social impacts of this. And people are using oak tree ring records as a way of trying to build out this climate record, and things like ice cores. And this is an example of one of these records, where you can see I've put in an estimated rainfall record. So this is a particularly bad time. You have a very bad moraine, notice the dead sheep, and this happens in 1428. And then a series of very, very dry years with very, very poor crop yields. And you notice the rise in prices. Look at the grain prices, it's inverted. This has gone really expensive because the yields are so low. And at this period of time, we report the lowest yields of wool that are ever reported in Britain. So it's a really, really bad time. And you can start to think about these impacts between the climate, then the production, and then the impact on energy, transportation, heating. Because these animals are stressed, you get an increase in disease. And also the people are stressed too, they get disease too. Then you get malnutrition, demographic growth, and then you get crisis. Then you have all these feedback through memories, and so all this interaction happening in real time in small parts of the country with different rainfall patterns. And so if we think about this in terms of what we could learn, we have got documents from all these manor court rolls from all of these places. Okay? Now remember, it looks like now, as of the last few days, we can start picking up the pathogens because they get much more intense at the points when you get these animals being very stressed like they are in this period. And you can then look, of course, these are very low resolution maps. We can get high resolution maps because we've got manor court rolls from across the country. So that's really cool. And then my student Sam, this is his slide, came up with this. So he just wondered, these sheepskin parchments are really fatty. If I can put solvent through the parchment and suck it through, I can collect the fats out of the parchment. Okay? If you get out the fats of the parchment, what can you learn? You can do deuterium isotopes. Deuterium isotopes are a signal of the rainfall, and this is our pilot data. So you've got here the instrumental measurement of this so-called year without a summer. This is the year after the eruption of Tambora. And you get this very, very strong climate signal. And we've compared, this is a reconstruction from all the different tree rings. This is the actual data in black here. And this is, we have predicted the rainfall just from the lipids extracted from the parliamentary rolls. And these are written at the end of the sitting of Parliament, and we can then back calculate from the data from this, is that this must be the water the sheep drank between the months of March and June. Okay? And what we're doing now is that team is now feeding sheep, or have just been feeding sheep on heavy water to try and track through the accumulation of the fats so we can really nail this down. In addition to this, incidentally, we can also see whether we have carbon as C3 or C4. Now, in England, that's not very interesting because C3 is the kind of carbon growing in plants in wet environments. But we're hoping, we've just put in funding to try and look at the Cairo Geniza, where you have the longest climate record in the world, the Nilometer. And we want to look at specific years there of drought and flood and link that to documents in the Cairo Geniza, again, dated documents. We're particularly focusing on the years just before the start of this new millennium. So like 900, 980, 990. So yeah, so if we can look at that, well, can we get anything else from this? I mean, we've pulled a lot already. Well, you've got salt price. Look at salt price jumping up. Can we say anything about salt? Well, yeah, those skins, when they're moved, they're salted, okay? If we have salt, and this is now, it turns out to be a huge story. In parts of the world where salt is very expensive, salt is too good to use on parchment. But here's a stack of skins in a parchment manufacturer, modern one. And notice some of them got a slightly red tinge to them. Talking to leather makers and parchment makers, they get a stack of these salted skins. And if they leave them too long, they start to go a slight purplish color, and eventually they go red, and they call them scarlets, and they throw them away. And so you can see here, again, this is Swedish silk prices in log scale going down. What we can see is that's a salted rawhide. This is going fresh material. This is not our work. And notice here, we start to get these very purplish spots. Those purple spots are caused by active biodeterioration of parchment, which has become more humid. And it's the organisms living on the skin of the parchment are basically revived and are destroying the skin. Those skins, when you analyze them microbially, we have a whole series of bacteria, and this is early, I've got better data than this actually, but what we're seeing is a series of halophilic bacteria. And what we see is greater diversity in halophiles in Spanish material, less diversity in British material, and no halophiles at all in Icelandic material. So you're seeing this difference in pattern. Now, this is very early days, but the idea is we can track in real time the change in the halophiles, and perhaps from that, we'll get a story about access to salt. And because this is only low quality salt that's used in parchment making. And then the thing that you probably thought we were doing is, well, what about the animals themselves? What about the genetics of the animals themselves? Well, so we've got these periods of time. This is a great period of time in the 18th century when they're starting to improve animals. And so the question that we started out with, which turned out to be about the most difficult thing we've done, we thought it'd be the easiest, most difficult, was to actually get out interesting genetic data from the parchment itself. And I think it's a wonderful quote that, the story of English sheep is not in the, it's been lost from the text. And what we've got is we've got it in the animals that were left today. And we have Robert Bakewell there on his horse, his improved horse. And he developed this thing, which is the New Leicester. And this is a failed experiment. He highly inbred his animals. And And so what we've been intrigued by is, I can't get my genetics colleagues to be that interested. I think I'm getting them interested now, is we can look at things like the wool quality in the parchment. We can see the follicle patterns, and this is work by Malcolm Ryder. And you can then compare that with what we see from, say, wool pricing. And then you can do, this is a search of texts, and you can see here's the New Leicester, this is Dishley's New Leicester coming up here. And this is the Dishley Society being discussed here. This is the society, and this is his sheep. And it crashes and burns very quickly. It's so inbred that what the New Dishley Society were doing was that they were loaning out the rams, but the rams were so inbred they had no sex drive. And the stories about these animals just sitting in the corner of the field, they weren't tapping any of the females. So the whole enterprise went down. He lost all of his money, end of the New Dishleys. And then suddenly you get this merino craze. These are the animals from Spain, which arise, and then they become hyper popular. They get crossbred with everything, and then they also prove to be a failure. And we haven't done any of the genetics work on this yet, but I think this would be a really fascinating thing to do because we have all of these data documents, so we can look at this in real time. And yeah, we can talk here about the fact that various. This is a wonderful thing called the reports from the Board of Agriculture, which is from 1793 to 1822, and they're documenting all of these experiments, so we can look at this question. And then this is a new data we've just got. So this is the first attempt. This is Matthew Teasdale's work to ask the question: can we postcode the parchment? And this is a question that most of my paleography colleagues want to answer. And the answer is, yes, we can, but we can only do this in those parchments with a very, very high PQI that have not been exposed to a lot of lime. The longer they're in lime, the more the DNA degrades. All that's left behind is DNA wrapped around histones, which is not very meaningful. It's centromeric DNA, it's structural DNA, so it's not useful. So now we at least have a way of saying that looks good for DNA and that looks not good. But what we can now do is we can look at the pattern, the origin of parchments, at least at the level of Ireland, England, Scotland, continental. That's all we can do so far. But yes, we can postcode the animals. And more than that, we can start to look into texts and look at books and say, who's related to whom? And so we can see in this particular text, those are two brothers, and they have got the same, they've got the same father and different mothers. So these animals must be from the same flock. And then this one is a cousin slightly further away. This is a single text here. But we've also got some folio in that text which are completely unrelated. They must be coming from outside. So you can start to see that they're using animals from the herd and they're using animals from outside. And as we start to do this, and these are in order in the folio. Again, here we don't see a very strong pattern of male and female, but you can start to pull apart the way the text is being constructed based upon the genetics. Okay, so we can now look at the relationship in the animals. What else can we learn? Well, so another interesting thing is foddering, because of course, if we're interested in the health of the animals, we're interested in those difficult climate years. What can we learn about the signals in the parchment? What can we learn about the animals and their animal health? So what we've done is I've been on eBay buying bits of parchment, and they're now free to anyone else to study. I've got about 600 pieces because I own them, so people can destroy them, right? And so what's happened, so this is a study where they use these dated parchments, they're all legal documents, all sheep, to actually track isotopic values. And this is very, very early days at the moment. But we've got some things here with carbon. There's a big change in carbon here. These are sheep that have lived on a C4 diet, on a diet which is associated with plants from much warmer places. So I've had a PhD student looking at all of the historical accounts records, and it turns out that what's happening here post the Napoleonic Wars is we are bringing a lot of animals in from Uruguay. These are probably animals from Uruguay coming into the record. This is bulk carbon. Bulk nitrogen, we're seeing some animals with very, very elevated nitrogen, but other than that, no obvious pattern, a huge range in values. But when you then look at the health of the animals, what's intriguing here is we've got animals in poor health which have got quite elevated nitrogen. And this is related probably to the remetabolism of their skin. And I've talked to somebody about this today. I'm really excited about this because there's some technologies out there that may allow us to do single compound amino acid nitrogen on these tiny fractions of parchment. At the moment it doesn't work, but I can see it then in a few years' time to get very high resolution data on these animals. And then the other question is, I'm talking to some others, what other markers might there be in these skins which might be markers of stress? Now, what else can we find there? Maybe there's markers of antibodies, who knows? I doubt it, but we know. So looking at these markers and using this kind of data, and all of this is physical bits of parchment that you can take away and destroy. Okay. So then we go on. Well, that was the parchment. What about the bindings that they're covered in? Is there anything special about those? I mean, I won't talk much about this because there's too much to talk about. But it's kind of, first of all, rarely do we get books in their original bindings. Sadly, they tend to get rebound. But here's a great story. I mean, what we notice in the foundational books for the monasteries of the Cistercian movement. I must introduce you, if you don't know, to Bernard of Clairvaux. And Bernard actually, his movement, it's a challenge to the Benedictine movement. They live in a much more with nature. They wear white vestments. What they mean by white is they're not stained. They're white. So white can also mean beige, the white vestments. And they have these hairy bindings. They don't look very hairy now. The hair's been mainly worn off. And they were thought to be possibly maybe smooth calf deer, and then maybe they were wild boar. So yeah, we looked at them. Big surprise for us. I think other scholars already knew this. They're seal skin. So it's intriguing. So this whole Cistercian movement, and we can start finding these seal skin now in lots and lots of covers of very, very important books in the Cistercian monastery. These seals, we think still, we haven't got the age of these animals yet, is under 2 weeks, which means they're white. We can then track them genetically, and they're coming from the northern coast of Norway and Greenland, right? So these are seals that are moving, then they're almost certainly moving with, it's a Viking trade, and they're moving almost certainly with this wonderful work done by James Barrett and colleagues on walrus ivory. And it looks at what's happening is that as the Vikings move to Greenland, they start to access the walrus ivory, and a secondary product, they're killing these baby seals. These baby seals are white-skinned, and those white-skinned books are taken by Bernard of Clairvaux to bind key documents in the monastery there, and the Cistercians rapidly produce new daughter abbeys, and those daughter abbeys do the same thing. So, for instance, the foundational document of I've got the name of the abbey now. They're wrapped in sealskin, basically. So you're seeing that these important documents are also wrapped in. They're copying that same idea. And we have the pictures we have of these seals. Most of these people had never seen a seal. The illustrations of sealskins, they look like dogs with fishtails. So, I mean, finally, if you keep going on, then what you can think is, well, we can see these pigments in the documents. We can also see pigments in the teeth of nuns. This is lapis lazuli. This is a great story led by Tina Warriner and Anita Rodini. And so are we actually seeing this link between the people and their documents? That's much harder for us to do. We have seen the presence of lots of salivary protein and lots of skin acne on those pages which are most actively engaged with. So, for instance, in the York Gospels, it's the book, the devotional page in which everyone kisses the Gospel. So we can see that. We can't see human DNA because it's just too mixed. But the idea of starting to think about the scribes that made it and link it to these texts is something we've been thinking about. And then in terms of practice, one of the things that I think is most intriguing, and I'm almost finished, I promise, is thinking about the dialogues of the Exchequer. And this is Bishop Richard Fitz-Neal. And what he says is that this is a very interesting text he writes to basically re-establish the Exchequer in London. And he says that you must use sheep because it's difficult to make an erasure. And we don't know quite the date, but it's kind of late 12th century. Then a student working in Paris with Anneliese has analysed Morimond, a very important daughter abbey of the Cistercians, and she finds this. This is the data from animal bones. There's a change in proportion. There's a bit more cow early on and more sheep later on. There's a change there. But notice the very large error bars because it's quite hard to date the bones. Then she looks at the charters. The chart, here's the charters. The dark colour are the internally produced, the light colour externally produced coming in. And at the same time, pretty much as we think that Richard of Sneel's writing, all the charters are on calf internally in Morimond, and then they switch and they're all on sheepskin. And that's happening in the Exchequer in London at the same time it's happening in a Cistercian, key foundational Cistercian monastery. So I don't know who's talking to whom and who makes the decisions, but we're now seeing a practice which is spread across Europe. And so that's me to finish. But one of the great things about this is the science is very straightforward. And the Novo Nordisk Foundation have done a wonderful thing in Denmark. We have created a laboratory, and we're not actually analysing parchment, we're analysing leather from the metro excavations in the centre of Copenhagen. There was so much leather discovered that school children are now coming in, they're learning zooms in real time, they're doing the analysis themselves, and we have the first paper where they identified the fact that they were using horse skin leather at a time when it was illegal to use horse skin leather. So, surprise, surprise, people keep breaking the law. And so, yeah, that's me done. So, yeah, thank you very much. And it's been a pleasure.